Good afternoon, everyone. My name is Tawny Samiski. I'm an entomology specialist with UMass Extension, and it is my pleasure to welcome you all to our invasive insect webinar series. This series is born from a collaboration between the UMass Extension Landscape Nursery and Urban Forestry program, as well as our fruit program. And it is made freely available to you through funding support by the Specialty Crop Block Grant Program at the U.S. Department of Agriculture. So we thank them for their support. I also have here with me today Jeffrey Jouet. And excuse me, Jeffrey, if you could please proceed to the next slide. Jeffrey's helping us with tech support in GoToWebinar. All right, so quickly before I introduce today's speaker, I'll go over some reminders about our pesticide and association credit process. So all instructions for receiving pesticide credit for today, Massachusetts categories 29, 35, 36, and the applicator's license or core license, as well as our association credits will be shared at the end of this webinar. So please remain on the webinar until the end to receive these instructions and to take a quiz for pesticide credit. This quiz is only required for those of you who are looking for pesticide credit. And if you sign off the webinar before taking this quiz, you unfortunately will not be allowed to retake it and will not be awarded the pesticide credit. So please stay with us through the end of today's broadcast. I also want to take a moment to remind our audience about the question function in GoToWebinar. So in your control panel, you will see an area where you can type in questions and send them to us throughout today's broadcast. And I will do my best to answer any that I'm able to answer, and I will save those that I cannot for today's speaker to answer after she is done presenting. Another reminder to folks that will be taking the quiz, if you are using a cell phone, just be careful of the back button. While you're taking the quiz, you may get kicked out during that process, which again, we don't want to happen, so you can get your association credits. All right, so without further ado, let me introduce today's speaker, and we can switch over to her presentation. Today's speaker is Dr. Julie Gould. She is an entomologist with the USDA APHIS PPQ, and she will be speaking to us today about progress towards controlling the emerald ash borer with biological controls. So please take it away, Julie.
Hi, thank you, Tawny, and thank you everybody for joining us in these absolutely bizarre times. Just so you know, if you hear dogs barking, it's because I'm working from home, so my apologies in advance if they start barking. I've been working on emerald ash borer for quite a number of years, and we've fortunately been making a lot of really good progress, and I'm going to tell you something about the pest and about the progress that we've made. Emerald ash borer was discovered in 2002, feeding on ash in southeastern Michigan. It was thought to come to the United States on solid wood packing material. We import a lot of goods from China, which is where this insect is from, and the emerald ash borer spends about 10 months of the year inside of wood, and so things are moving so fast, it doesn't take very long for a tree to get cut down, made into a pallet, shipped to the US, and then thrown out behind somebody's business. And the emerald ash borer came out and started a population. So a typical life cycle, and this will become important later on in my talk, this is a 1-year life cycle I'm going to talk about, but sometimes when it's colder or other circumstances, the emerald ash borer can take more than 1 year to develop, and that's actually important to the story of biocontrol. But if it does do a yearly development, the adults come out in June. They feed for quite a while. It's called maturation feeding. They need to get nitrogen to produce eggs. They will then mate on the bark of the tree. The female lays her eggs in crevices underneath the bark. These are showing some of the eggs. You can see they're very flat because they are laid between layers of the bark. The larvae then penetrate through the bark and feed between the layer of the bark and the wood. They feed in the phloem and cambium layers. And once they have completed development, they go into the wood and make overwintering chambers. And you see this larva is folded up into sort of a J shape. We call those J larvae, and that's also going to be important to our story. In the spring, they will form pupae inside of these chambers in the wood, and then they emerge in the spring and they chew D-shaped holes. This is very diagnostic of beetles in the Buprestid family, and so they all chew these D-shaped exit holes when they come out of the tree. It's not the adults that cause the damage, it's the larvae. The larvae produce these galleries, and when there's lots and lots of these galleries in the phloem layer of the tree, it cuts off the flow of nutrients, and it will eventually girdle and kill branches. And it starts at the top of the tree. First you get death of branches, and then the entire tree dies. And hundreds of millions of trees have already died due to emerald ash borer in this country. And it's got quite a lot of impacts, ecological impacts. Ash isn't the most common tree, but often where it occurs, it's in sensitive riparian areas, and where it occurs, it's a high proportion of the forest. And you can see that this is a riparian area where many, many of the trees have died, and they've all died from emerald ash borer. And then there's economic loss. Ash is a very popular street tree, and many, many of those have died around the country. They make many products from ash, baseball bats being a big one, handles for buckets, many other products. And it also has cultural and social implications. The native peoples make black ash bark baskets that are very beautiful, and they're important to their economic as well as cultural heritage. So the big picture, it's about 2% of the country's total leaf area is ash. It costs about $300 million a year to manage this pest. And we expect $50 to $60 billion in ash-related economic losses nationwide, and then $282 billion lost from timberland. So big economic impact. So when the insect first came into this country, we found it in 2002, but it had probably been here since the early 1990s. They wanted to eradicate it. You always want to eradicate something if you can. And so they tried mechanical control, and that basically consisted of finding infested trees, cutting them down, and chipping them up into small pieces. And also regulatory. Don't move firewood. Don't move logs. Don't move nursery stock with ash. So, and they also did some research on chemical control. Chemicals are really, really effective against the emerald ash borer in an individual tree. They actually, especially emamectin benzoate, when it's injected into a tree, works really well, but you can't use it for a forest. And so, unfortunately though, for the eradication efforts, EAB has a huge potential for spread. The adults are strong flyers for sure, but it's really artificial spread that just got this all over the place. Infested nursery stock. In the very early days, there was a nurseryman in Michigan who ignored the quarantines that were in effect, and he sold nursery stock to Maryland, and there we were. We had EAB spread halfway across the country overnight. Excuse me. Infested firewood is a big one in the early days too. If we wanted to find emerald ash borer, we literally would just go to campgrounds. That's where we looked. as people were moving firewood and moving emerald ash borer along with that firewood. And of course, infested logs is also a way for them to spread. So this is the current known distribution of emerald ash borer. Every time I give this talk, I have to get a new map because it's just moving very quickly. You know, just a couple of years ago, we had holes in Vermont, we had a hole for Rhode Island. It had jumped out to Colorado, and so South Dakota, Nebraska, Kansas, Oklahoma were, you know, not infested, but it's really filled in over the years. And so pretty much the entire United States east of the Rockies is infested with emerald ash borer. So I'm going to back up just a tiny bit and talk about what is classical biological control, because as you can see from that previous slide, we're not going to eradicate emerald ash borer. We need other tools to control this pest. And what I do is classical biological control, which is the importation of specialist natural enemies for the sustained control of a previously introduced pest. The very first case of classical biocontrol in the country, I will talk about that in a minute. I got ahead of myself. So the benefits of classical biocontrol is that it can be very, very specific. So some pesticides and herbicides are more general and can kill many pests, but biocontrol agents very often are parasitic wasps or parasitic flies, and very often they will attack only one species, only the target. So they can be very specific. They can be self-sustaining because these insects are reproducing on the pest and going on to then attack more pests. So it's a self-sustaining. In the long run, it can be very cost effective, and it can be extremely effective. So there are risks, of course, to classical biocontrol. The one that most people are the most concerned about is possible non-target effects. Is it, if we release something against the emerald ash borer, will it attack our natives? Will it attack the bronze birch borer, the two-line chestnut borer? There are concerns about negative impacts to humans. I don't know if you all have heard about the multicolored lady beetle. It hides out in people's homes in the winter. It was released as a biocontrol agent, and it actually causes allergies. Some people are allergic to this biocontrol agent. Biocontrol is not easy or quick to implement. It takes time, and it never will completely eliminate a pest. It will reduce the density. And the other thing is the upfront costs can be considerable. It costs lots of money. So because of these disadvantages, you really need to make sure that there's a high probability of success before you pursue the development of biocontrol. This was the example I was going to talk about. It's the cottony cushion scale. It was the very first biocontrol project undertaken, and it was undertaken in the 1800s. This cottony cushion scale was not native to California, but it almost destroyed the citrus industry in the late 1800s. And so scientists went to the native range of the scale and imported this very adorable beetle called the Vidalia beetle, and a parasitoid, this little wasp, and it was highly successful, very, very successful. And since then, there have been a lot more projects, and there have been over 5,000 introductions against arthropod pests worldwide since 1889. Cassava Mealybug is another very big success story. Cassava is native to South America, but it's very important in Africa. Sometimes up to 70% of a person's daily energy intake will be from cassava. And in 1970, the Cassava Mealybug from South America was introduced into Africa and caused up to 80% crop loss. But scientists went to South America, where the Cassava Mealybug is from and where it's not a pest, and they found this little wasp. They introduced it into Africa, and it was very, very successful. And they estimate that the benefit to cost ratio was 500 to 1. In other words, and And every year the benefits just keep accruing because the success just keeps going year after year. This is another ash pest that I worked on when I was starting out my career. I don't know if you all know, I actually got my PhD at UMass under Joe Elkington many, many years ago. And I worked at my very first job after graduating was to work on the ash whitefly in Southern California. And this was a big pest. Whiteflies are sucking insects and they produce honeydew, which gets all over the leaves and causes the growth of this sooty mold. And the pest populations in Southern California just exploded. And we went to the native range of the ash whitefly and introduced this very small wasp, and it absolutely did a number on ash whitefly. Nobody to this day knows about ash whitefly because it was just such a success story. So overall, there have been many examples of successful classical biocontrol. Only about 16% of them have provided complete control. Another 42% have provided partial control. Some of the projects that didn't work failed because they were very limited in duration or in resources. It really does take some effort to get biocontrol into place. But when it does work, the cost ratios can range from 3 to 1 to over 100 to 1, and the benefits continue to accrue over time. In Australia, scientists did an analysis of benefit cost ratios and found that for biocontrol, the benefit cost ratio was 10.6 to 1 compared with 2.5 to 1 for pesticides. So overall, the benefits of the successful projects outweigh the combined costs of the unsuccessful projects. So if it seems like a good target pest, it's good to proceed. So when do you use classical biocontrol? Well, and let's see how that applies to emerald ash borer. When the species is not native, check. Established for at least 5 years, check. Causing economic and ecological damage, check. And when eradication is not possible, absolutely. It was just moving way too fast. So we decided to pursue biocontrol of emerald ash borer. And these are the steps that one has to take in order to do biocontrol. First, you want to study the biology of the invasive pest. You need to know what you're up against. You want to survey for natural enemies. You don't want to release exotic natural enemies if there's something here that might do the trick. There was not for emerald ash borer. There were less than 1% of EAB were attacked by anything. Then you conduct foreign exploration. You have to select potential agents. Not all agents are amenable for biocontrol. Then you have to bring them into quarantine and conduct host specificity testing. Lots and lots of paperwork. You have to justify what you're going to do. And then you need to apply for and receive permits. You then have to get them out, mass rear them in large numbers, which, as you can imagine, is difficult for a pest that lives under the bark of a tree. And then you have to release them. And then you have to evaluate the efficacy and study the impact of non-target insects. So we have done all this, and that's what I'm going to be telling you about. And it's been a lot of work, and it's been what I've been doing, keeping me off the streets for the last 15 years. As I mentioned, we did do a survey of natural enemies in Michigan, and less than 1% parasitism. We do see these insects cropping up once in a while, attacking EAB, but nothing that causes enough mortality to provide control. So we started surveying for natural enemies of EAB in China. We surveyed between 2003 and 2007. These are some of my collaborators. In the slide on the bottom left, that is actually Fraxinus pennsylvanica. That's green ash, and it's growing in China, and it was fruiting and reproducing in China. And these are some of the insects we found, the parasitic insects that we found. This is Tristracus planopenisi. it's not a very big wasp, but it still has an ovipositor that drills through the bark, and it lays its eggs inside of the emerald ash borer. If you follow and look at the top picture, that is actually, if you look at where the arrow is, you can actually see the larva of the parasite inside of the EAB. It's what's called a gregarious parasitoid. So there's many of these little wasps growing inside of that EAB. We actually call them swimmers. That's the technical term. So they start out inside of the EAB. If you follow along on the slide in the upper right, that's actually an EAB that is just chock full of these parasitic larvae, which then, continuing on, burst forth into the gallery. They then pupate. They have a naked pupa. They don't spin a cocoon. And they do this 3 or 4 times a year. So each wasp is producing 50 to 100 offspring in a single EAB, and they're doing that 3 or 4 times a year compared to a EAB which takes 1 to 2 years to develop. So they showed a lot of promise. This is Spatheus agrilli, which was found further south than Tetrasticus. It was found near Beijing, China. This is a picture, a very nice picture of the Spatheus agrilli ovipositing through the bark. They actually lay their eggs on the outside of the EAB, and they paralyze the EAB first, so it is rendered immobile. The larvae then feed on the outside of the emerald ash borer, and then they spin cocoons inside of the EAB gallery. They also are gregarious, although they only produce maybe 8 to 10 cocoons per EAB, not like 50 like Tetrasticus. And but they also have several generations per year. And then finally, we found a egg parasitoid. This thing is minuscule, and I was very happy I didn't have to work on it because it's very hard to see. It's called Oobius agrilli, and it lays a single egg inside of an EAB egg. And then you can see we've removed the parasite larva from the egg. This is the larva and the pupa. And on the bottom is a dark EAB egg. That's an egg of an EAB, and when they're parasitized, they turn black, and you can see the round hole that the adult wasp chewed out from. Now, when I said that you don't want to consider all natural enemies, this is one that we found that we did not consider ever importing. It's called Sclerodermis puparii. It was found in Tianjin, China, near Beijing, but parasitism levels were low. A high percentage of the females were wingless, which means they wouldn't be very good at dispersing. And members of this genus have been known to sting people. So we decided that this one was not very high on our list of potential good biocontrol agents, and it was not worth pursuing. So we left this one alone and never considered it for biocontrol. So our first step before release is to do testing in the lab to see if the parasitoids are specific. Do they only attack emerald ash borer? So we tested bronze birch borer, two-line chestnut borer. We tested other things that get in ash, like Neoclitus, the red-headed ash borer. We tested a lot of native species, and we found that all of the that Tetrasticus, Spatheus, and Oobius all attacked significantly more EAB than non-target insects in both choice and no choice tests. We found that we did some testing with Spatheus agrilli and found that in an olfactometer it was attracted to ash leaves but not to other plants. We looked in China at other members of the genus Agrilus. We never found the 2 larval parasitoids on anything else other than EAB in China. And we found that there is a Spatheus native to the US that once in a while attacks EAB, but no native Tetrasticus are ovipositing on EAB. These things have not switched hosts, and we did not expect our Spatheus or Tetrasticus to switch hosts. So this is just a big slide of all of the paperwork that we had to do: Fish and Wildlife Service, environmental assessments. We had to post on the Federal Register, lots and lots of paperwork. But we went through all that, and we did get a federal permit issued, and we started releasing in July of 2007. So we were releasing small numbers because I can't rear very many EAB. And so the USDA APHIS set up a very large mass rearing facility in Brighton, Michigan. And this is not an easy pest to rear. I mean, the first thing you have to do is you have to get EAB eggs. Well, we don't have an artificial diet for emerald ash borer. So what we actually would do is we would let Mother Nature rear them for us. We would go out into the forest, cut logs containing EAB pupae, warm them up in cardboard tubes. The adults would come out. We then put them in these little cages with ash foliage. We actually had the ash foliage. There's a tree called evergreen ash, and it's grown in California and Florida and Hawaii, and we would have the leaves shipped in to feed the emerald ash borer adults. And then they actually we put screening and coffee filters on the top of these little cages, and the adults would stick their ovipositors underneath the screening and lay eggs. We then took those eggs and wrapped them around a cut ash log, which we then kept in water, and the larvae would develop normally in these logs. We then took pieces of paper with eggs on it and gave it to Oobius agrilli, or we took these logs with the larvae and gave them to Tetrasticus and Spatheus. So that was how we continued to rear these insects. And originally we were actually releasing adults, but we decided to take advantage of the fact that we're rearing pupae, and we now release the Oobius agrilli in the eggs in these little pill bottles. We call them oobinators. That's the technical term. And then we release the larval parasitoids in ash logs that contain the pupae that are ready to emerge. So we developed a mass rearing techniques, and you can see that we've been rearing over 1 million parasitoids per year, which, if you think about it, is absolutely astonishing. We peaked at production of Tetrasticus in 2015 because the next year in 2016 started producing more Oobius, and you can only produce 1 Oobius from 1 emerald ash borer, or you can produce 100 Tetrasticus. So it's kind of a trade-off. So that's why production fell. It wasn't because we lost our capability or anything. So we have released parasitoids in 25 of the 35 infested states so far. Some states are just newly on the list, and we haven't found good release sites yet. So now that we've gotten them released, we've been sampling to see if they've established. And you can peel trees. That's one method you can use. You can peel the bark off of trees, find the larvae, see if they're attacked. We also peel the bark and sort it and sift it to look for emerald ash borer eggs to see if they're parasitized. And you can rear logs and bark in tubes and collect the adults in those little cups. But this has been our best tool. It's called a yellow pan trap, and we put them out on infested trees, and the insects collect themselves in the little yellow bowls. And that's been a very effective way of finding, because you've got to know, if I release these things, are they actually persisting in the environment? Tetrasticus planopanisi has been recovered in 17 states so far. This is another really nice picture of a larva packed full of parasite larvae. This is pretty impressive. We have recovered Oobius agrilli in 14 states, and both of those species are persisting, and we're finding that they're moving, they're dispersing. Spathius agrilli was recovered. It could reproduce and overwinter, but it has failed to establish. We've never to date seen a reproducing population of this species. And climate matching, if you look at the yellow area, this is the best part of the United States that matches up to Tianjin, China, where Spathius agrilli is from. And this is not where we were releasing. We were releasing these things in the gray area, where they are not expected to, where the climate is not as good a match. So we decided to conserve resources and release Spathius only south of the 40th parallel. But the problem is that even though Tetrasticus is establishing well in the north, it has a very short ovipositor, and it's restricted to very small diameter ash trunks and branches where the bark is really thin. So we needed something like a Spathius, and fortunately, foreign exploration was continuing during this time, and we found this species called Spathius galeni, which has a long ovipositor. It can drill through the very thick bark, and it's from Russia, Vladivostok, Russia, which climate, you can see the yellow portion on the map, is much more similar to the northern US. And we found it to be quite species specific, and we got unanimous support for our release petition, and Spathius galeni has so far been recovered in Colorado, Connecticut, Delaware, Illinois, Massachusetts, Maryland, and New York. So we are starting to see some good establishment of this species. But the big question was, why was Spathius agrilli not establishing in the north? And would it do better in the south? And would Spathius galeni be any better in the north? So we undertook some experiments in Tennessee and New York to try to tease out the life cycles of these parasitoids and how that relates to the life cycle of the EAB. And we found that in the south, in Tennessee, the majority of EAB are overwintering as J larvae. Remember, I showed you they were down in that overwintering chamber, and they're very deep in the wood, and they're not available to parasitoids. EAB populations in New York have remember I was talking about, some have a 2-year life cycle. So in New York, you have all stages of larvae under the bark available to parasitoids. And in this study, we found that Spathius galeni and Tetrasticus both emerge early in the spring, when hosts are available in the north, but not available in the south. And this is why Spathius agrilli was not able to survive in the north. It emerges later in the year, and it's synchronized with 1-year EAB populations. In the north, it actually emerges too late in the fall and has a dead-end generation. So what we expect then is that Tetrasticus planopenisi and Spathius galeni would establish in the north, and Spathius agrilli would establish in the south. And we actually did a huge nationwide study. We had 92 sites throughout the country looking at where EAB overwinters as larvae. You need larvae, not J larvae. And we were able to create this model and this map. The red dots show where Tetrasticus planopenisi has established. The white dots show where Tetrasticus has not established. You'll see the white dot up in Dalton, Massachusetts. And I swear to God, Dalton is a black hole for Tetrasticus. We have released tens of thousands and no establishment. It's like the Bermuda Triangle of biocontrol out there in Dalton. Anyway, we did this model, and we looked at the growing degree threshold. And so what's going on here is that the further south you go, the hotter it is during the summer. And the hotter it is, the faster the EAB develop. And when they develop really fast, they become J larvae and they become unavailable. So if you look at the left-hand column, that's the percentage of EAB that are predicted to overwinter as larvae. That's what you want. So in the north, 51 to 80%, you want that. That's a lot of larvae. And if you look at the right-hand column, the percentage of sites with Tetrasticus establishment, 92% of them had establishment. That was that one Dalton site where we didn't get establishment. So that's what you want. So by developing this, we can show land managers who are thinking of considering EAB, they can look at their growing degree numbers. And this is growing degree days between January 1st and September 30th. And they can say, how many growing degree days do I have? What is the predicted percentage of EAB? What does that mean in terms of establishment? And should I release Tetrasticus in my area? So this is a tool that we developed for the biocontrol practitioners. So the question is, though, is this working? And the answer is yes. I'll give you the answer up front. We did some long-term studies in Michigan where we had released Tetrasticus oobius and Spathius. And this shows what happened to the EAB density. The study started in 2008. There was this huge peak in 2009 where we had 40 to 50 EAB per meter squared. These are huge numbers of EAB larvae that we were sampling. The numbers of EAB then declined and have remained so ever since. Even though this only goes up through 2014, I can say that the density per meter squared of live ash phloem has stayed down. Woodpeckers are a really important mortality factor. Woodpecker predation, even as the density of EAB was falling, woodpecker predation remained about 40%. So that was a very important mortality factor. Also, during that time, though, we saw this increase in parasitism by Tetrasticus. They also were contributing more and more to the mortality of EAB as time went on. So EAB density peaked in 2009, and we got a 5-fold decline by 2014. We were seeing increases in parasitism by both native parasitoids and Tetrasticus, which helped reduce the EAB density. Tetrasticus is now the dominant parasitoid. And we still haven't really evaluated the impact of oobius because honestly, it's just really, really hard to find. But it's certainly there and it's reproducing and spreading. Spathius galeni is also doing a really good job. This is a busy map, but it just shows on the top Spathius galeni parasitism. You can see it's even greater than Tetrasticus parasitism, which is what's shown on the bottom. So we think that this is going to be a really important contributor to mortality of EAB in the future. And this is really important. We are not going to save big ash trees. We just cannot. We are releasing a few thousand parasitoids against millions of emerald ash borer, and the big trees just die. Even if we release parasitoids, the big trees succumb and die. What we are really targeting is the next generation of ash, young ash trees. And this is a study that we did in New York, where we had been releasing parasitoids for many years. We had been releasing them since 2012, and we conducted this study in 2017. And this is a pie chart of saplings. There were 120 saplings in Randolph, New York, and you can see that 64% of them had no EAB in them at all. 17% had some old galleries, but they had recovered and there were no new galleries. Only 18% of these young ash trees had any EAB living in them at all. And this shows the mortality. This is at 2 plots, Hudson River Valley on the left and Randolph in the spring. And look at the 4th instar. This is the instar where the parasitoids attack. And if you look at the graph for Randolph, New York, we sampled 120 saplings. This was 20 saplings each at 6 sites. We found 0 living emerald ash borer larvae. Most of them had been killed by Tetrasticus, and a lot of them had been killed by woodpeckers. So the parasitoids and the woodpeckers are doing an amazing job at keeping populations of EAB down, because the densities of EAB at these sites were mostly below 1 per meter squared, very low density of EAB. So the one final thing I would like to talk to you about is some work that we're starting to do to save some of these big ash trees. As I mentioned, the big ash trees die, but we also have emamectin benzoate, which is really, really good at controlling EAB in very large ash trees. So I asked the question, can these systemic insecticides save these trees, keep them alive while the parasitoid populations build up, disperse, and establish? And will the suppression of the EAB populations allow the parasitoids to increase more quickly relative to the pest? And can we eventually stop using these insecticides because the biocontrol agents and the woodpeckers will cause sufficient mortality? We think this might work because Tetrasticus and Oobius are establishing and dispersing. Insecticides are very effective at keeping ash trees alive, even when there's a lot of pressure from high density EAB populations. And EAB populations reach a peak and decline at the same time that percent parasitism by native parasitoids in Tetrasticus and Oobius are increasing. So, and woodpeckers continue to find and remove about 40% of mature EAB larvae, even as the EAB density declines. So we had some hope for this, and we decided to do some studies of EAB in urban forests. And we established 3 research sites: Boulder, Colorado, Naperville, Illinois, and Syracuse, New York. And we found that we indeed saw, the map shows where we were finding Tetrasticus by year. Even you can see this is Syracuse, New York, and you can see the 3 yellow dots at the top. That was actually where we released the parasitoids. And you can see a light blue dot at the bottom of the top circle. That's over 2 kilometers. That was within the release year. I mean, even within the year of release, those things had moved 2 kilometers. And by 2017, they were, the control plot was what was at the bottom. And by 2017, they were down in all over the control plot. And you can see that the numbers are increasing. And we didn't have Spatheus galeni to release at the beginning of this study. We released it later, but we have also found it in all 3 of the study areas. And so far, we've only found Oobius in Colorado, but we found it in large numbers. In 2019, EAB densities were way down, parasitism was high, and we have actually stopped our insecticide treatment. We are going to see, we're going to keep monitoring, and we're going to see if we can continue to get control of EAB without pesticides. This year, we're actually extending that study to natural forests. Ash seeds do not live long in the forest, only a year or 2. And we need reproductive ash trees to restart populations in the forest after the EAB populations collapse. So we are actually treating large trees in the forest and releasing parasitoids just like we did in the urban areas. And as I've mentioned, black ash is very important economically and culturally, and it's very susceptible to EAB. So we are going to concentrate on some black ash forests in northern New York and northern Wisconsin. So in summary, our early results are promising. Tetrasticus and Oobius are establishing and dispersing. We are continuing to release Spathius agrilli in the south and Spathius galeni in the north. EAB and young ash trees are suffering high mortality from woodpeckers and Tetrasticus. Tetrasticus and Spathius galeni are establishing and spreading in urban areas and can contribute to IPM in urban areas. And biocontrol is complicated, requires time and patience and research, but we are hopeful for the future of ash in our forests. And that is all.
Excellent. Well, thank you so much, Julie. And if you want to leave your PowerPoint up, if you need it while you answer questions, we'll certainly leave the controls with you during this period. So I have a couple of questions from the audience. The first is from Gary, and he asks, when do the EAB eggs hatch and when are the larvae feeding?
So EAB, it depends on where you are. Normally, EAB adults first come out when there's 450 growing degree days, base 50. So around here, that's sometime in June. They then do this maturation feeding for a couple of weeks. So you're getting into egg laying maybe the beginning of July, and the eggs will hatch in about a week. You know, again, it all depends on where you are and what the temperature is. And then the larvae will continue to feed, and larvae that get enough heat units will become J larvae and enter the chamber. Those that do not will simply become larvae and spend the winter under the bark as larvae, and then they will continue feeding. You know, we have had up in northern Michigan, we have collected adult beetles in August, like the end of August. So, you know, when do the eggs hatch? It depends on when the adults come out. And the further north you go, the more spread out things are. And you really could find eggs throughout the summer.
Thank you. Another question from, I'm not sure, this might be a different Gary, but will the emamectin benzoate treatments also kill the parasitoids? Which maybe
That's
you
the
answered
beauty of it.
earlier.
Absolutely not. Because the parasitoid, for one thing, Oobius is attacking the eggs under the bark on the outside of the tree, and emamectin benzoate is a systemic, so it never comes in contact with it. So Oobius is fine. The other thing is that the emamectin benzoate kills the larvae, and a dead larva will never be attacked by a parasitoid, because that's actually how the parasitoids find the EAB. You have to wonder, how do they know where the larva is under the bark? And what they actually do is they hear the EAB larvae feeding. The larvae make noise as they feed, and the parasitoids can hear that through the bark or feel it, you know, as they're walking around. And so if there's a live larva in there, it means it has not been killed by emamectin benzoate. And I'll tell you, if a larva gets into emamectin benzoate, it dies. It's very deadly. In fact, we had, in our urban IPM studies, we found larvae in some of these treated trees, because, you know, the uptake of emamectin benzoate is not perfect. And we found 5 EAB larvae in these treated trees. 3 of them were attacked by Tetrasticus. So they're totally compatible, which is excellent.
Great, thank you. Another question from Alicia. Please give an example of a cost ratio 500 to 1.
Well, I think that, you know, the Cassava Mealybug that I talked about is certainly a cost ratio, you know, benefit cost ratio, because if you mean, explain what that means, it means the cost, all those things I explained to you about biocontrol is the cost of the foreign exploration, the cost of doing the testing, the cost of creating doing the paperwork, and the cost of the rearing and the mass rearing and the cost of release. Those are all the costs. But the benefits are accrued because you're not having to treat with pesticides, you're not having crop loss, you're not, all these costs associated with treating the pest. Those are the benefits that you accrue. And once classical biocontrol is in place, you continue to accrue those benefits forever, pretty much. I hope that answers the question.
Thank you. And if not, Alicia can send us more detail in the questions area. I have another one from David. We are treating trees with emamectin benzoate every 2 to 3 years in Massachusetts. How will we know when we can back off our treatments?
Well, that's a really good question, because unless, we're not doing any of these biocontrol releases, at those sites. And until the parasitoids are established and you really can't back off those treatments. Somebody needs to be releasing the parasitoids, making sure they're establishing before you can stop treatments. And, in Massachusetts, I would actually love to work with some of the cities and towns in Massachusetts. I had actually reached out to Ken Gooch before he retired and said, Ken, let's do some of this in Massachusetts. And I also reached out to my sister who lives in Cumberland, Rhode Island, and she contacted, her people, and nobody got back to me about doing this. So, if anyone is interested in learning how to do this and how to release the parasitoids, and how to monitor for them, I'm happy to advise and happy to help.
Thank you, Julie.